Hi, I'm Amanda McGill Johnson with the Nebraska Coalition for Life-Saving Cures. We're an organization that supports, promotes, and advocates for health science research. And we're here today to launch our new Talking Research program, which is a Lunch and Learn series to help Nebraskans learn about the various types of health science research happening around our state. One of the things that I've heard a lot about is that folks might know what's happening at the universities in their own cities, but maybe not what's happening in another part of the state. So I'm very fortunate today to have Dr. Kimberly Carlson, a professor and assistant chair of biology at the University of Nebraska at Kearney, here to join me today. How are you, Doctor?
I'm doing great. Trying to stay warm. We got a little bit of snow last night, so
It
had
is
some had some faculty that couldn't get in who live outside of Kearney, but not as bad as the last snowstorm we had.
Oh my goodness. And we're about to hit that cold front. So I don't think any of us are looking forward to that. Now, Dr. Carlson is here today to talk about her research with fruit flies. And I think I don't speak just for myself when I'm saying, what the heck do fruit flies have to do with our health as human beings?
Yes, that's a really great question, and it's interesting how fruit flies became the model. They are actually the model for all genetic studies. When you talk about an invertebrate model, um, it happened a long time ago, but as we started studying fruit flies, well, when you look at human disease and human health, studying humans is not fun. Okay, they bite, they yell, they hit. I was fortunate enough to be able to do human research when I worked at the University of Nebraska Medical Center, doing HIV research and volunteering, doing some work with the Alzheimer's disease research study group. And I realized at that time, as much as I like people, I really don't like doing research on people. And so when I started my research, I wanted to use a genetic model that's easy to manipulate, but that actually shares a lot of similarities with humans. And you're thinking, no, we don't have wings, we don't fly around. No, that's true. But we have a lot of the same genes. And a lot of the most important discoveries we learned about humans and human diseases, we first learned in fruit flies because they're very easy to manipulate. We can take a gene out, we can add genes in very easily. They make large numbers of offspring in a very short period of time. A female will lay 100 eggs in the morning, in the afternoon. They'll just keep laying eggs all day, and they're to complete their life cycle. It's 24 hours, or excuse me, 2 weeks, 22 weeks to get to where they can start producing large numbers of offspring. In the wild, they live for about 25 days on average, just because you got wind and predators. But in the lab, we can have them live from 50 days to 100 days and keep doing that. So they're a great system for us to use.
So you can see relatively quickly how altering a gene in one generation impacts 3 generations down the line.
Yeah. So, for example, we can alter a gene. We can do that in two different ways. And both ways are making genetically modified organisms. We can actually use very specific molecular biology techniques to manipulate a gene. And that's relatively easy with fruit flies. You can make different little constructs of DNA and inject it into the fruit fly egg, and voila, there you go. Another way is if you're talking about CRISPR-Cas9, which is a new technique that's out there, you can just feed it to the fruit flies and it makes the changes. Okay, can't do that to humans, but we can do that to fruit flies. Another way is by doing selection experiments. So my major background is in aging research, and so we like to see what's the trade-off between having an increased lifespan and any other function. So what we found is if you take flies and you age them and only allow the oldest ones to reproduce, and then out of their offspring, you only take the ones that are the oldest ones, allow the oldest ones to reproduce and keep doing that, you can extend the longevity of fruit flies from 25 days to 140 days. So we can manipulate their genetics in multiple ways in the lab.
Huh. So how does that then translate over to us? Like you said, you can't feed us some of these changes, but in
Sure.
terms of that longevity piece, even.
Sure. So we've learned a lot, especially about metabolism and immunity from fruit flies. We can find, so in humans, when we do things like, let's say, we want to figure out what the genes are that underlie obesity. Okay. We can do screens in humans. Okay. They're called genome-wide association studies, big word. All it means is we take a lot of people and look at their genes. You get these candidate genes, and you're going, well, I can't take and remove this gene from this person to see what happens. But what we do is we see, does the fruit fly have a similar gene? If they do, then we can study that in the fruit fly. We can learn how to manipulate it. We can develop drugs. We can do pharmacogenetics. We can make pharmaceuticals, treat the fruit flies and see what the response is before moving up into like a lower vertebrate, a mouse, and then move up into humans. So we've done this with a number of different genetic diseases, including our current coronavirus pandemic that we have. We've been testing drugs for humans on fruit flies.
Really? I had no idea.
Yes. Yes. So hydroxychloroquine that everybody, talked about the miracle cure, uh, really isn't. But what we found out is that chloroquine can actually be used to treat, to help alleviate some of the symptoms, the long-term symptoms, long-term after effects that we get from coronavirus infection, that it can actually relieve those. And we discovered that in fruit flies.
That's really cool. Tell us a little bit about what your specialty is with this fruit fly research.
Okay, so I started my fruit fly research looking at aging. I'm very interested in the genes that underlie aging. So what I'm really interested in is, we do really well. We're going really good, and then we hit, in my case, my 40s, 50, and now I'm 50. Right here, and all of a sudden you start going. So I'm really interested. What is this? So a few years ago, my students and I, we did a project. It was called a large cage study. So we used tens of thousands of fruit flies. And we took time points for their whole lifespan, which was about 72 days. And we looked at all the genes at every time point to see how they're changing. And lo and behold, what we found was that as your immune system changes, you die faster. So
Mhm.
your immune system is very tightly linked with how long you actually live, which makes sense. Okay. So when I was at UNMC, I worked on HIV. I went from being a fruit fly geneticist to a human geneticist, somehow got there, I don't know, and then back to being a fruit fly geneticist. So one other thing that came out of these large cage studies that we did was my I have lots of girls that always work for me. We had them in these large cages, and you have to clean the cages. And the flies were collected about 18 months apart. And when you clean the cages, flies live in close. They live in their feces. So they're cleaning the cages. You take an ice scraper, clean the sides. Well, they come running down to my office, and they're so excited with two containers of feces. One came off in rolls, nice rolls. The other one just crumbled. And I'm thinking these girls are crazy. And they said, we think there's a difference. And I'm like, well, obviously there's a difference. What do you think it is? And so they characterized every bacteria and every virus that was in the feces. And the difference between the two was a virus called Norovirus. And Norovirus is a new virus. It was first discovered by our Swedish colleagues. And they named it Nora, meaning beautiful. And a lot of people confuse it with Noro, the cruise ship virus, not the same.
Mhm.
It's a corona-like virus in the same family as polio virus. Okay, we can't get it as humans. It replicates in the gut, and for the longest time, people said there's no effect on the fruit flies. They have no phenotype. Okay, you can't tell that they have it. I had a very brilliant undergraduate student, Abby Rogers, and she was taking another class, and she said, "I have to come up with my own project I can do statistics on." She said, if this is like polio virus, maybe they don't move as well because, you'd see people with polio, have braces on their legs. And I said, well, that's an easy test. And lo and behold, after 3 years of getting all the data together, the girls, these undergraduate girls in the lab, discovered that norovirus does cause a locomotor defect. They move slower. Well, and overall, what might that mean? That might mean that this virus is causing the flies in their brain, they don't really have brain, causing them to have neurodegeneration similar to Parkinson's disease or Alzheimer's disease. So now we have a model. We use this to study poliovirus. It's also an RNA virus, just like SARS-CoV-2 is. So we can learn about um, RNA viruses. We can also we've made virus-like particles. We could actually make a vaccine to our norovirus in fruit flies. So it gives us a lot of different information on, something that's potentially related to a human virus, and we learn tons.
So it sounds like students play a really important role in the research that you do.
Yeah, at UNK, we have a huge emphasis on experiential learning and undergraduate research. We really want our students to get that experience, whether you're a biology major, or you're an art major, an English, or a teaching major. Everybody has the right to do a project and get some hands-on experience. My undergraduates, and every once in a while, I have a good graduate student. They're my colleagues. I always tell them I work for them. Okay, I'll go down there. They keep the lab running. They keep the research running, and they're really enthusiastic about the problems that we're working on. We have a new project that we're working on that I think is really cool. I might have a new graduate student. We're looking at how we can use lipids to make fruit flies become more heat tolerant. And how does that affect humans? Well, think about sub-Saharan Africa. It's very, very hot. If we could alter the diet of the people down there and make them so that they can tolerate the heat more, they'd actually live a more productive life. And with global climate change going on and temperatures getting warmer, if you could, alter your diet so you can, handle the heat better, it'll be better overall. So that's one of the new projects we're starting.
That is fascinating. I had no idea that diet could potentially impact our ability to withstand temperature.
Yeah, so it's your genetics, and not just your genetics, it's also your epigenetics. So above the genome, it's all those tags, that you have on your genes that turn your genes on and off. And the best way to explain that is every cell in your body has the same genetic makeup. We have the same genome, whether it's your head or your feet. So if you ask yourself then, if that's true, why do I have my eyes here? Why aren't they on the top of my feet? Right? It has to do with how the genes talk to each other in these tags. The tag says, ooh, it's time to turn on now and make eyes up here. And those tags are different down in your feet that say, nope, no eyes down in the feet. So all that stuff makes us who we are. And it's highly easily to be manipulated.
What is it like on a day-to-day basis to be working with such small little creatures as fruit flies, as those things that most of us see as pests?
Oh yeah, exactly. And people do, because I get a lot of calls. I've got fruit flies. How do I get rid of them?
Mhm.
And actually that's very easy. But for me, when I was a graduate student, I was looking at the relationship between reproduction and aging. And reproduction is very, very mechanically and energy expensive to the female. And so I would dissect the females, every single female fly, and I pull out their ovaries and dissect apart their ovaries, because they're like this, and they have these things called ovarials. And
Are you using, like, what tools do you use to do that?
yeah, so we have micro dissection tools that we use, and my PhD advisor said you should be a microsurgeon. You've got great hands. So they're very fun for the questions. So I have students, we have a paper I just submitted before I came on to this talk, looking at the gut microbiome and how the microbiome is affected by norovirus and flies. Of course, that tells us a lot about our own microbiome. And the student dissected out dissected out the guts of these flies, and then the gut has 3 parts, and so she dissected the gut apart and did all her studies. You have to have really good hands to do it, and my students are fabulous. We have contests to see who's faster, and I don't have the dexterity anymore to do it as fast as they do. So but it's great because we don't have to get any external approval to do it. There's no if you work with animals, you have to get IACUC approval. Humans, you have to get IRB approval for all the ethics, make sure everything's great. Nobody cares about fruit flies. Okay. And you can just squish them if they get away, and we have, lots of different strains, and the kids that work with them, you either love them or you hate them, because they go home with you in your hair and stuff,
Oh,
you swallowing them. But they're actually great. When we sex them, the most basic thing we teach people how to do is tell the sex between the male and a female fruit fly. And the kids get so good at it that they go into the grocery store and want to land on their finger and they're like, yep, that's a female. And they're telling people. So
You can tell that easily.
yeah, yep, males have what are called big black butts. So the females have a pointed abdomen and the males are rounded and they have a black tip on it. So all the kids that take genetics here at UNK, they all have to do fruit fly lab for about part of the semester, and they all could tell you the difference between a male and a female fruit fly. So everybody gets the experience if they're a biology major or a pre-health major at UNK working with fruit flies.
Fascinating. Are there other bugs that have been as helpful
Well,
to research?
sure. So genetics, of course, Drosophila is because it's so easy. You put them in a bottle, you put them in a vial. You can store them on the countertop. We have special rooms, but other people have used. So let's see. There is mosquitoes. There's a certain type of mosquito that people use. Of course, that's good for looking at disease transmission. Cockroaches is another one. I can't stand cockroaches, so I can never work with them.
Most of you.
Silkworms are good. They have a great immune system. Butterflies, sometimes people work with. The real difference between using a fruit fly, and there's 1,600 Drosophila species. Melanogaster is the number one model species, or genus. Yeah. But there's a lot of those too. So if you have bigger fruit flies or littler fruit flies, but yeah, there are some other ones, and people use them. I don't like mosquitoes because they bite. I don't like cockroaches because they're cockroaches. Butterflies get too big. You have to have more room. And fruit flies are they're really easy. They're really easy to work with.
And in terms of the genetic similarities between us and fruit flies, are they one of them that has more similarity than any of those others?
Yeah, so fruit flies have about around 1,400 genes that make proteins, where humans have about 24, 25, or sorry, they have 13 to 14,000 genes. I'm sorry. Humans have about 25,000 genes that make proteins. Of those, we share 60% of our genes with fruit flies. We have 60% of the genes are similar. But of disease causing genes, it's 75 to 80% that are the same. That's what makes them such a wonderful model. So, especially when you're talking immune system genes, most of what we've learned about the human immune system, at least in innate immunity, we learned from fruit flies.
That's fascinating. And I believe when I was doing research, even for this, a lot of Nobel Prize winners, their research is directly linked to this.
6 Nobel Prizes using Drosophila. The last one was in 2017. The one that I'm most excited about, though, was when Jules Hoffman won the Nobel Prize for his work on innate immunity, which is the area I work in. And his direct collaborator is my direct collaborator on Norovirus. So
Oh,
I would say
cool.
I'm twice removed from a Nobel Prize
At
because
six
of my
degrees or
collaboration.
2 degrees of separation.
That's right. That's right. That's right.
I want to just mention that if anybody's out there, folks are watching this live, if you'd like to put any comments in, we'll questions in the comment section, please feel free to do so. What first really got you interested in working with fruit flies?
So I was an undergraduate student here at UNK, and in my genetics lab, we worked with Drosophila, and I really liked working with them because they were easy to manipulate, easy to use, and I found them fascinating. And we take them at different stages, and we, dissect them apart and look at their chromosomes, and I thought that was fascinating. And my research project was actually working with a fish, and I had worked with fruit flies, and then I was working with this fish, and I'm like, they're bigger. I mean, they're a little fish, but they're bigger. Their eyeballs were hard to grind up just all these things. And so when I got my PhD, I knew I wanted to do it in genetics. And I was fortunate enough to start rotating in a lab with a person that worked on the genetics of aging using fruit flies. And he's very famous in the Drosophila world. And he was wonderful. And I just like them. I mean, like everybody else is complaining, I have to write this protocol to be able to use my this, and I'm like, yeah, I got fruit flies, and they're easy. They're so easy to work with, and we use ether to anesthetize them, and some people are like, oh, it's the ether you like, and I'm like, well, Sure, why not? Okay. I can't even smell it anymore because I've used it so much it's deadened all my senses. So it's really, I mean, they're just a great little model. I find them fascinating because, humans, we have 23 pairs of chromosomes. Drosophila have four, and the fourth one doesn't even really count. So, but think about that. They have so much fewer chromosomes. Theirs are much bigger than ours. But you know, we don't have twice as many genes as they do. And we have 75% of the disease causing genes are the same. So I just find that fascinating. And you can manipulate them however you want. And I find that fascinating too.
That's, yeah, that's really awesome. Where do you get your fruit flies then? Are you just kind of reproducing them or do you bring them in from somewhere? How does that work?
Okay, so we have a couple different stocks that we use. So we got for the stocks for our norovirus flies that we use. It's a stock that was created by Dr. Dan Haltmark in Sweden, and he made it for his norovirus studies and the other immune studies that he does. And it's a special line of flies, and so he gave those to us, and we keep those. And he has since retired. He still does stuff, but is retired. And so we are the stock house for that line of flies. If anybody wants them in the world, they come get them from us now. The other ones, there is a stock center. We have stock centers for Drosophila work. The great thing about being a Drosophilist is we like to work together. We are very open. It's very different than the HIV world that I was working in. In the Drosophila world, we share. We like to share. I just got an email from a new colleague friend that I made at Harvard University. She's going to start looking at the Rolovirus in some childhood disease. And she asked me if I could send her fruit flies, which I will do. I'll send her fruit flies, my fruit flies. But otherwise, we can buy them from the stock center. We just started a project. We want to see so SARS-CoV-2 jumped from, we believe, a bat to humans. So we wanted to see, can our virus go into different species of Drosophila? So melanogaster is where we know it's at. So we got species of Drosophila from around the world. So we have a species from England, we have a species from Brazil, and we have a species from Africa. And now we're seeing if the virus will cross into those other species. So there's all sorts of stock centers.
Fascinating. What would you say, getting into maybe some fun questions,
Okay.
the biggest pros and cons are of working with fruit flies? Because clearly you're passionate about it.
Oh, yeah.
Some of the things you love about them.
Oh, so pros, I've listed a lot of those. Cons. So for ours, when you're talking about development, you have to keep them at a very specific temperature. And so we have incubators. So I have a number of incubators and I have special rooms. And over one break, I believe it was Thanksgiving, I came back and you try to keep them right about 25 degrees Celsius, which is room temperature. I came back and my incubator was at 45 degrees Celsius. So all my fruit flies were fried. Now, being the smart person that I am, I keep them in multiple different incubators just in case. So, I mean, that's always discouraging because you are dependent upon temperature. And if you have too many, you can get mites, and mites are fun to look at, but they ruin your whole project. You can have cross contamination with norovirus in the flies. It's so highly infectious that if you get one fly that has it, it can take out an entire stock that you have. I mean, we can get rid of it because we can take the little tiny fruit fly eggs and take off their outer coat and get rid of all the virus, but it really is time consuming to do that. So that part I don't like, but the rest of it, there's really not another. I've worked with mice. I've made transgenic mice. I've worked with humans. I've worked on other organisms. I help with crickets and grasshoppers. I just really like fruit flies. They're easy. They're easy to use. And the community of people is just fabulous that we work with.
You had mentioned earlier that people reach out to you with how to get rid of fruit flies. Go ahead and give us the inside scoop on the easiest way to do that if you have them
Okay.
in your home or wherever you may.
Sure. So inevitably, you'll come home with them from the grocery store. And when you come home with fruit flies, the easiest way to get rid of fruit flies is to get a container. It can be a coffee cup or whatever. Fill it about a third of the way full of red wine. And then you put in a little bit of table salt and a little bit of dish soap. So Drosophila melanogaster is actually the vinegar fly. Okay, so they like wine. Okay, so they'll go in, they'll drink the red wine, they'll get a little drunk. The they're terrible alcoholics. They're used in alcohol studies too. The dish soap will get on their wings and they'll be too dense to fly, and the salt keeps them on the top of the liquid till they die. So basically, they drink themselves to death. Now in the lab, we don't use red wine. We do use just water with salt and dish soap, but we directly put them in there. But that's the easiest way to get rid of fruit flies. And you just set it out on your counter and it'll be full of fruit flies. And that's the easiest way to get rid of them.
Somebody made the comment, don't give the flies the good wine.
Oh no, actually you want the worst stuff you get. The funniest thing is, that old saying, you catch more flies with honey than you do with vinegar. I had a couple students, an undergrad and a graduate student. They were both, the undergrad was going to be a teacher, he is a teacher now, and the graduate student was a teacher, and they wanted to do this project. And so we looked at it, and nope, you catch more flies with vinegar, your wine, than you do with honey. So yeah, they love, and vinegar even better. Take some white vinegar. If you don't have red wine, if you're not a red wine drinker, I'm not, I just take vinegar and set it out and do the same thing. And they love it. They won't get drunk, but they'll drink enough vinegar and get enough soap on them that they'll just die.
That is very helpful. Can you use, this is another pre-submitted question, but can you use CRISPR to get a giant fruit fly? Seriously, okay. Do you use CRISPR in your research? I think you mentioned it a little bit.
Yeah, we do use CRISPR. So for those of you out there listening, CRISPR-Cas9 is a new tool that we have in our toolbox that was first found in bacteria that we can use as a geneticist. The one thing we always wanted to do was be able to find a gene, shut it off, and see what it does. Now, the tools to do that weren't available to us till very recently, within the past decade or so. And even more recently, CRISPR-Cas9 allows us to do it in humans and all different organisms. And we do it in fruit flies. So can you make a bigger fly? The answer to that is yes. Now, will you make a big fly? No, because physiology restricts how big you can make it. So I used to watch the old horror movies, and it'd be like the giant locust or the giant praying mantis. Or if you watch Godzilla, Mothra, or even Godzilla herself, I think, they can't get that big. Their physiology is going to restrain them. That's the only problem. They're going to be, restrained by their own physiology.
So we're not going to have fruit flies as dog type pets anytime soon.
Oh, and what a filthy pet that would be.
Oh dear. Well, we've reached about 30 minutes now. Is there anything final you'd like to share, or something else interesting about fruit flies? Well, call it a day.
The one thing that I said early on is that we are using, we use Drosophila. We have models of Drosophila for everything. We have it for Alzheimer's disease, Parkinson's disease, HIV, any human disease that you can think of, we're using fruit flies for. But I think, to me, what's most interesting right now is the work that we're doing with SARS-CoV-2. And we learned how, and I find this fascinating. We know that SARS-CoV-2 enters cells by a very specific receptor. So a virus needs a receptor to get inside the cell. The receptors are called ACE2 receptors. Now, interestingly enough, mice don't have those receptors. Okay, so you can make a transgenic mouse using CRISPR to have those ACE receptors. But Drosophila have a very similar receptor, and you can give them the SARS-CoV-2, and then you can take your pharmaceuticals, your vaccines, and you can try it on fruit flies before you try it on humans. And so we've made progress, and there's a new pharmaceutical that is going to be going through the system for COVID-19 that was first found in Drosophila.
Wow, that's so cool and fascinating. We did get one question popping in, if you have a few more minutes.
Sure.
Just how similar are the lab flies to the wild fruit flies?
Oh, that's a good question. So lab flies are pampered, okay, because we take care of them, but they're very similar. So if you collect melanogaster out in the field, it is just the same with a little less genetic. Its genetics are a little bit more wide open. So lab flies are inbred, very highly inbred, because we just keep them in a bottle and they just keep breeding with each other. But they look exactly the same. And fruit flies are beautiful. They can get beautiful wing patterns. Their eggs, they have appendages on the egg. So egg looks like an oval with these, we call them wings, but they're appendages. Different species have more appendages. They do different things and they're beautiful. But the wild type are just like the lab ones. We like to go to, and the fun thing about collecting wild ones, go to a vineyard, okay? And we like to go to the vineyard to collect fruit flies. Now, our vineyard that we tried to go to was Max Creek Vineyard in Lexington, because the person that owns it was a faculty member at UNK, and the student that wanted to be a teacher, he worked there and he was friends with them. The only problem, we love going out there and we use every excuse to go out there, but they are so clean, they don't have any fruit flies.
Oh, sure.
They have a very clean facility, no fruit flies. And if they do, they're like, hey, you want to come out here? And then they know how to get rid of them. So yeah, the wild type caught ones look just like the ones in the grocery store. In fact, we go to the grocery. We've done experiments where we go up to the grocery store to collect fruit flies. So you can get them anywhere.
Wherever you can get them. That's cool.
Wherever we can get them, we go for them. But vineyards are the best places to go.
Cool. Well, thank you so much, Dr. Carlson, for spending some time with us today and doing our first, being our first talking research guest. You know, as I said, she's from the University of Nebraska, Kearney. We're getting some comments thanking you for this great presentation. I ask people to keep watching back on our Facebook page for additional talking researches and presentations in the near future. Thank you. Take care and stay safe.
All right. Thank you. Bye, everybody.